Hello friends, good morning. Welcome to some biology class. Now we shall discuss about the structure and function of thymus. Thymus is a primary lymphoid organ. Its main function is to facilitate the development and maturation of T lymphocytes. It resembles a thyme leaf and hence the name. In human being, thymus is bilobed. It is located just behind the top of the sternum below the thyroid glands. Thymus is a derivative of pharynx. It develops from the epithelium of the third and fourth pharyngeal pouches. In human being, at the time of birth, it weighs about 15 to 20 grams. Thereafter, it increases in size up to a weight of 40 to 50 grams, after which it atrophies by a process called involution. In adults, it is really hard to distinguish it from the fat in which it is normally embedded. In birds, it consists of 7 lobes on each side of the neck. Thymus is a pyramid-shaped lymphoid organ. In human being, it consists of 2 lobes, left lobe and right lobes, which are merged in the midline. Thymus is protected by a dense connective tissue capsule which penetrate into the interior at several places and form septa called trabeculae. Each lobe of the thymus is organized into many lobules, which are separated from each other by septa. The tissue of thymus is distinguished into an outer zone called cortex and the inner medulla. The cortex mainly consists of thymocytes and epithelial cells. Thymocytes are T cell precursors. They are supported by a meshwork of finely branched reticular epithelial cells, which is continuous with a similar network in the medulla. Cortex is characterized by the presence of heavy concentration of thymocytes. It is the principal site where thymocytes proliferate, differentiate and mature to become functional T lymphocytes. Proliferation of thymocytes is evenly distributed throughout the cortex. After maturation, T lymphocytes migrate from cortex into medulla, from where they enter into the bloodstream via medullary vein, thereby adding to the T lymphocytes in the peripheral blood and lymphoid organs. In addition, the cortex also contains thymic nerve cells, which are large epithelial cells having their own nucleus. These nerve cells are known to internalize thymocytes through extensions of their cell membrane. Their cell surface and cytoplasmic vacuoles express MSC class 1 and class 2 antigen. The interaction of these antigens with the developing thymocytes determine whether the thymocytes will undergo positive or negative selection. The thymus also consists of macrophage, dendritic cells, a small amount of B lymphocytes and neutrophils. Thymus differs structurally from other lymphoid organs in that it do not have lymphatic vessels draining into it. Blood vessels enter the cortex via septa near its junction with medulla. The medulla consists of vascular structures, reticular epithelial cells, scattered lymphocytes and interdigitating cells which interact with reticular epithelial cells. In addition, the medulla also contains Hassel's corpuscles, which are small masses or wholes of epithelial cells around a central core of degenerating epithelial cells. Their function is not known. Thymus is a primary lymphoid organ. Its principal function is proliferation, differentiation and maturation of T lymphocytes. Friends, this is all about the structure and function of thymus. For more updates, please subscribe my YouTube channel, Sam Biology Class.